So we are past time already. Algae. So algae are photosynthetic eukaryotic organisms found in aquatic environments. Algae are photosynthetic, which means they can what produce their own food through the help of sunlight. They can what undergo the process of what photosynthesis. So they are also what's called true cells. That is, organisms with what true cells. Now, algae, they are what primarily found in aquatic environments. They are found what primarily in water environments. Though some of them can be found in moist soils, but the primary location of algae, in case you see it in your exam, is what aquatic environments. Now, just know this one too. Algae, they lack what a true stem. They lack a true leaf, so they are not classified as plants. They are classified as protists. So
Classified
key
as what?
features:
Huh?
protist, P-R-O, protist, Protist,
TIT.
Tist.
Okay.
All right. So key features: they contain chlorophyll. Chlorophyll is what green coloring matter found in plants that enables them to carry out what process of photosynthesis. So they are classified as what chlorophyll. Sorry, they contain chlorophyll. Two, they can be free living, that's unicellular. Sorry, they can be unicellular or multicellular. That is, they can. Please, someone's mic is on. They can exist as what single cell or what in colonies or in multiple cells. Now, very important very important part about this algae is that they would provide oxygen. They provide oxygen. There was this fun fact I read one time. It said that algae, they provide more than 50% of the world's oxygen supply. Algae alone, they tell us that I think around 70% of the oxygen supply is from water, right? Now, algae, they are what's found in aquatic environment. They are what produce 50%, more than 50% of the oxygen supply. So if algae should go extinct, my dear, I don't know. I don't know what will become of, but God will provide a way. All right. So they provide oxygen, and number two, they form the basis of aquatic food chain. There is no reproduction here. They reproduce by both sexual and asexual means. Just write it down. They reproduce by both sexual and asexual means. You can also write they are motile organisms. Some of them have flagella and can move around. So this is an algae. So the last type we have here is called archaea. Archaea. Some call it archaebacteria. So
I
the
didn't
whole
see any slide for that alga. Initially, you brought it out and then you took it away.
are you seeing this now?
No, not this one. I mean that alga we just finished now.
Yes. This is slide.
Not this greenish something. The right
Which one?
up. I need to snap it. Uh-huh.
Okay. You want to start the right up?
Is it where it started?
Yes.
Can I,
It's very small. The other ones were from my head.
pardon?
Yes, it's just this. Have you snapped it? I think the slide should be sent to you guys, okay? So don't bother. I will have to tell your coordinator to send the slides. I will send it to him too. So, okay. Now, first of all, they are unicellular prokaryotes. They are unicellular. They are single-celled prokaryotes. They don't have true nucleus. Okay? So just by applying these terms,
Please,
they are unicellular.
sir,
Sorry? Eh?
excuse me, sir. Please, the algae. Let us have the slide of the next. subtopic you are into.
Are you seeing my screen?
Yes, I can see your screen, but it's alligator that is still on mine.
It's not algae. This one is Ake. Archaea.
No,
No, sir, the
I know
algae is not showing. It is the
it's
greenish
not showing.
algae that is showing. It is not
It's
showing, sir.
alligator that is showing.
Okay, so what are you seeing now?
The
So
greenish
basically, what do you want?
algae.
You
The
want the write up.
algae,
You want the
algae.
write up. You want to see the write up.
Yes.
Yeah,
Write up for algae.
Yes,
Algae,
the alkaline
algae,
characteristic.
alligator,
Okay,
not
yes.
are you seeing it now? Are you seeing
alligator.
it now?
No.
No,
You're not seeing the write up.
We are not. We
Yes.
are
We are
no.
only seeing
No.
the that okay
Yes,
now.
yes.
We
We
are
can
seeing
see
it
it
now.
now. Yes,
You
yes.
are
Thank
seeing
you,
it
sir.
now.
Yeah, thank you,
You are
sir.
seeing it now. Wow, I'm
Yes,
actually
sir.
in
Yes,
another
Yes,
sir.
sir.
I
slide.
can see it now.
Okay, so let's continue then. So, okay, there are unicellular prokaryotes. You are still seeing it, right?
Yes, sir. Yes, sir. We can.
Yes, sir.
All right. So there are unicellular prokaryotes that are genetically distinct from bacteria. First of all, they are unicellular. Secondly, there are prokaryotes. I told us pro means what? Pro means what? First. Karyote means what? Nucleus. But in this sense, though, it's talking about what? It does not have a true nucleus. It's like the earliest form. Hmm? So it does not have what? A true nucleus. They can also call it what? Post karyote. So an algae is what? A unicellular organism without true nucleus. But we have mentioned earlier about bacteria. We say bacteria are also what? Unicellular organisms without true nucleus. But now, the key word there is that algae are genetically distinct from bacteria. Something in their structure is different from that in from those in what um from those in what bacteria. Time is already against us. We have like this should be time for question and answer, but let's keep going though. Now,
Let's keep
key
on.
fe- all right. So I'll mention the key features, then I'll also okay. I think I included it here. Now, key features. Number one, okay, they would thrive in extreme environments.
Sir, you didn't tell us that distinct feature that distinguish it from bacteria.
Don't
You said
worry.
that.
Don't worry, calm down, okay? When I'm going forward, I'll tell you. It is there already. They lack peptidoglycan in their cell walls, okay? What they have in their cell wall is pseudopeptidoglycan, glycoprotein, and what? Polysaccharide. So don't worry, when I'm going, I'll be mentioning them, okay? So I don't want to jump from one point to another. So we what? Have a structured knowledge, understanding about it. Are we together?
Yes,
Okay,
yes.
Yes, sir. We're together.
all right. Thank you very much. So, key features about Ake. Now, they thrive in extreme environments. They are hard men. They live in environment that defies all odds. Now, that environment where no other organisms live, that environment where no other organisms survive, that is where you go and you see Ake. So that's why it is debated as what the oldest cell. That's why many people are saying it must be the what earliest form of cell. Are you with me? So why? Where they will find them is what? In extreme environment, something like a very hot environment, like a volcanic eruption. Those archaea are called is those archaea are called thermophiles. I think you should write this one down. That thermophiles, they are found in what extremely hot environments. Another type called halophiles are found in what in an environment that has high salt concentration. Halophile, HALOPHILE, halophile. Now, another another type of adaptation
The
is called
thermophyte is what?
eh?
Halophyte. You said that
They are
thermophyte is that fight in
thermophile.
hot environments.
That yes, exactly.
Fight
Thermophiles
in-
are found in what extremely hot environments.
What?
Halophiles are found in what in environment that has high salt concentration. I think question you should ask yourself is that how is high salt concentration a bad environment or a very harsh environment? Can anyone attempt? Anyone?
Alophiles is a harsh environment because most alkalis are not normally found in salty concentrated places, because when they are found in concentrations with high salinity, they tend to
Yes,
lose their water to the environment and they become plasmolized.
exactly. Very nice. Thank you. What's your name, sir?
Joshua.
Joshua. A very nice reply. Thank you very much. So, cells, number one, cells exist in isotonic environment. Isotonic means an environment that has what? Normal cell concentration both in the extracellular fluid and intracellular fluid. So there is no osmosis going on, or everything is going on as what? Equilibrium. Hmm? Now, if you should put a cell, okay, please know this thing I want to tell you. If you want, you can write. If you don't want, just learn it. You can see, you will see it in your exam. It's a very simple question. You can see red. Okay, let me take it structure by structure. Now, I told us that there are three types of concentration of salts. Number one is called an isotonic environment. In an isotonic environment, are we still following? Are we following?
Yes, yes.
Yes, we are
Yes,
following.
Or should I start from the scratch? Okay, let me go back. Let me go back. Now, a cell has an internal environment called the intracellular fluid, composed of the cytoplasm. Hmm? So the cytoplasm forms the intracellular fluid of a cell. Now, outside of a cell, the cell has a plasma membrane, a cell membrane, just like a wall that would separate it from the environment. So everything outside the environment is called extracellular fluid. Extra means outside. Extra. I want us to be learning all these origin of words. Extra means outside. Extraterrestrial, outside of land. Are you with me? So extracellular fluid means fluid that is what outside of the cell. Intra means inside. Intracellular fluid inside the cell. So um every other fluid outside is called extracellular fluid. Every other fluid inside is called intracellular fluid. Are we still together? Now, if okay, now what is what salt concentration? Now, the concentration of the salt, sorry, concentration of the environment, what? Oh God, I don't want us to miss this thing. When you hear salt, please, salt does not mean only sodium chloride. Salt does not mean only table salt. Salt means what? Can anyone attempt? Okay, let me just say salt is a compound formed when what? An acid reacts with what? A base to form salt and water, right? Okay, so you can have sodium chloride, potassium chloride, and the rest. So this determines how concentrated. I don't want to go so deep, so we'll not get lost. Okay, let's just be going with the little one we know. Just know
We won't get
salt
lost. Just
concentration.
pour out everything.
Okay, no problem. I love how you guys are going about this class. All right. Now, when the salt concentration, let's use a very common one, sodium. Now, sodium chloride, when sodium dissolves in water, it forms sodium ions and chloride ions. Chloride ions are what? More outside, right? They are more outside. Potassium are more inside the cell. I think in your second level, second year, if you are pursuing a medical course, med lab, or the rest, they'll teach you guys this thing I'm teaching you now. All right. So inside the cell you have more potassium, outside the cell you have more sodium, but in general we should still call them just salt concentration for simplicity. Okay, so when the concentration of ions inside is much, you say that intracellular fluid is what concentrated. At that state it is called a hypertonic environment. Hope I'm not mistaken. Now, outside, when the salt concentration outside is more, when salt concentration outside is more, sorry, it is called a hypertonic environment. When the concentration of salt outside is small, it is called a hypotonic environment. All right, environment. When salt concentration outside the cell is what? High, it is called a hypertonic environment. When it is small, it is called a hypotonic environment. Now, if you should take a red blood cell and put in a hypertonic environment, that is in an environment with low salt concentration. When you take a red blood cell and put in a hypertonic environment, an environment with high salt concentration, water is going to move out from the cell into the environment, causing what? Plasmolysis, just like Joshua stated out. That means shrinking of the cell wall and cell membrane. When you should take a red blood cell, any question? When you take a red blood cell and put in a hypotonic environment, that means environment is low soil concentration, water will rush inside the cell. The cell will now swell up and will now burst. It is called hemolysis. Hemolysis simply means what? Breaking. All right. Okay. So let me simplify everything I've told you now. Red blood cells, when placed in hypotonic environment, will rupture. Red blood cells, when placed in hypertonic environments, will shrink. This is just the point there, okay? So this is what you see in your exam hall. Probably question 1, a red blood cell was placed in a hypertonic environment. Which of this is likely to happen? Option A, it will shrink. Option B, it will rupture. Then you'll be see other stupid options. So you should know when placed in hypertonic environment, the cell is going to shrink, also called plasmolysis. When placed in hypotonic environment, the cell is going to swell up and eventually burst, also called hemolysis for red blood cell. So back to where we were. I was mentioning adaptations. We talk about, because I don't know where I was. Okay, let's continue. Now, Ake told us that they thrive in extreme environments, that is hot environment. I told us they are called thermophiles in spring and salty lakes. In salty yes, that was where we are. Environment with high salt concentration. They are also called halophiles. Also, there's another third adaptation. There is this type of bacteria that are found in environment with high acidity. Those ones are called acidophiles. They are giving us three now, right? So let's continue with our features. Number 2 feature, they lack peptidoglycan. This is one of the words, something that is distinct from bacteria. Bacteria has peptidoglycan, right? This guy, they lack peptidoglycans in their cell wall. So what they have in place of peptidoglycan, they have pseudo, that is PSEUDO. Growing up, my other sister used to call this pseudo. So they have pseudopeptidoglycan. Pseudopeptidoglycan. Please, not pseudo. All right. So they have method is just get your spelling correctly. Pseudo, PSEUDO. Pseudopeptidoglycan, or glycoprotein and polysaccharide in their cell wall. So this is one of the things that make it distinct from bacteria. I think, there are still other more. You can browse them up. Also, they have a unique biochemical pathway. This is another distinct something from the bacteria. They make use of histoproteins. All right, Melo?
This is
All right.
saying, are they not in the slide?
Are you seeing this? Are you seeing the screen?
I'm not. It's that first screen you shared is still here.
Are you seeing the screen now?
The first one you shared, that is what is here.
Tell me what you are seeing.
All right. Let me go straight to my question. You said that they lack peptidoglycan, but they have pseudo peptidoglycan. There's another
Yes.
thing you said after this one.
Okay. They also have glycoprotein. Glycoprotein. Please, if you're not seeing my screen, tell me, okay? Because it's just to help you guys. Glycoprotein. And the last one, polysaccharide. Okay, there is a very important point. This point is not in the slide. That is, okay, they don't cause disease. They are non-pathogenic. Unlike every other microorganism that cause diseases, these ones they are what non-pathogenic. Written.
Yes.
So let's move on to the next point, to next subheading. Okay, we have mentioned all these things. Aseptic technique. Aseptic technique. So are you seeing my screen?
Yes.
Yes, sir, I can see it.
You are seeing aseptic technique overview, right?
Yes, sir.
Yes, sir.
Okay, beautiful. So aseptic techniques are methods used to prevent contamination in microbial cultures. What this simply means is what there are what set of procedures, there are processes, there are rules you have to follow to make sure that what contaminations are not introduced in your culture. Culture, how will I best explain culture now? Now, culture means God, help me, help me. When you talk about culture, is a very common word in med lab scientists. It's just like what trying to create another environment to recreate what an environment in probably a test tube or a petri dish. When you talk about culturing a microorganism, it simply means you are trying to what grow that microorganism to create an ecosystem what in a petri dish. Okay, so one very one very thing about microbiology is that we are trying to very accurate as much as possible. So if you are to say that in this culture, what we have here, in this our small ecosystem, what we have here is let's use for example staphylococcus, that's a type of bacteria. You should not come there and be seeing penicillin. Penicillin is another is what penicillin is a fungi. You should not come there and be seeing HIV or be seeing plasmodium. No. So how can we make sure that no other microorganism will enter into this environment except this my one we are studying? So all these things are called what? Aseptic technique. It is just what? A means of procedure, a laid down guidance, just processes that what you should carry out. Are you getting me? So let me go again. There are practices performed under conditions designed to minimize what the entry of unwanted microorganism into a what sterile environment. So these techniques ensure that sterile conditions during experiment and laboratory works are kept. So let's look at the common aseptic techniques. Number one, sterilizing tools using autoclave or dry heat. Autoclave means heating with what? A little moisture. It's a type of heating technique where water is touched in, moisture is touched in what you want to heat, and then you heat it up. It's also called wet heating. So you sterilize your tool by what? Heating them through autoclave or dry heating. Number two, cleaning surfaces with disinfectants. Someone sent a question. Please use your voice notes. I can't leave this screen I'm sharing now, right? So, working near a Bunsen burner flame to prevent airborne contamination, using sterile tools and avoiding direct contact with open cultures. Please, this last point is very, very crucial. Avoiding direct contact with open cultures, especially for those aspiring to be med lab scientists. It's somehow a risky profession if you don't follow all these laid down rules and regulations. You can easily get infected by what? A culture you are preparing. Now imagine you are probably growing a culture of blood sample from a HIV patient, and probably you not cover up yourself properly, or you expose yourself, or you have a cut or something. You can easily get infected. As simple as that. So you use what? Sterile tools, and also avoid direct contact with what? Open culture. Please, you should know this by now. You don't talk, you don't chit chat in your laboratory. They're also part of all these rules and regulations. You should not even think of eating in your laboratory. I did that one day. I entered the cadaver room. Cadaver room is just like a mini mortuary. But in this time of scenario, we are not keeping dead bodies. We are operating on dead bodies. They'll give you your tools, surgical tools. You'll be cutting up dead bodies. Are you getting me? So you enter the cadaver room, and I was eating chin chin there. Thank God our instructor did not catch me. You'll be seeing dead corpse, everything there. I was busy eating. I was trying to form big guy. I'm not afraid. Oh God. University life is sweet. Don't worry, all of us will get there in Jesus' name. So, handling microbial cultures. Please always wear your gloves and your lab coat. Now your white overall, always put it on. Sterilize all equipment, either autoclave or dry heating, before and after use. Minimize exposure to cultures. And lastly, and very important as well, dispose of cultures and materials in appropriate biohazard containers. Dispose them properly. Maybe now you cultured what a HIV, or okay, let's use a very common something that is not that bad. A plasmodium, that's disease that the pathogen that causes malaria. You cultured what plasmodium. It's not when you want to throw it, you just come into the public and probably throw away the petri dish. A child or someone might not mistakenly go and touch it and can easily get infected if the person should move the hand to his mouth, to eyes, to nose, or any open parts of the body. Hmm? So we are expected to dispose them off properly in biohazard containers. So we have reached the end of today's class. Any question?
Sir, I have a question.
All right, I'm listening.
Yes, sir. When you were explaining these homophiles under Archaebacteria, you said holophiles are divided into three. We have isotonic, hypertonic, and hypotonic. Sir, please, can you explain what isotonic is again?
Sorry, halophiles. No, I'm not saying halophiles are divided into three. Please, okay, don't get me wrong. Halophiles are a group of organisms that can exist in high salt concentration. High
Okay,
salt
sir.
concentration means hypertonic environment.
Okay.
So I simply mean halophiles exist in hypertonic environments. So okay, hyper means high, tonic means salt, environment means your
Yeah.
what outside surroundings. So halophiles exist in what environment that has high salt concentration. So I wanted to explain to us what salt concentration mean. That was when I introduced 3 types of environments based on salt concentration, where we now have isotonic environment, hypertonic environment, and hypotonic environment. Isotonic means balanced. The salt concentration outside and that one inside are what equal. So there is no movement of water. Hypertonic environment means what? Salt concentration is higher. So osmosis takes place. Movement of water from what? A region of what? Low concentration to a region of what? High concentration. Right? So water moves from the inner cell that has low. Am I too fast?
No, sir.
Are you with me? Okay, are you with me back?
Yes,
Thank
sir.
you.
But too
So
fast, sir.
so okay, let me
Yes,
slow
sir.
down. Let me slow down. All right. So hypertonic environment means what? An environment with high salt concentration. Hypotonic environment means environment. Hello? Yeah, anyone answer the question? Okay, let me continue. So there was I hypertonic. All right. So now when you place a cell in a hypertonic environment, what happens is that osmosis take place. Water from the cell which has low salt concentration will now move to the part that has what high salt concentration, right? And I defined osmosis again. Osmosis means movement of water molecules from a region of low concentration to a region of high concentration. So when you place a cell in a hypertonic environment, water moves out. Are we together? All right. So now hypotonic environment means low salt concentration. Hypo means low, tonic means salt. Are you with me? So salt concentration. I think tonic means concentration. Sorry. So hypotonic concentration means what? Low salt concentration. Now, when you put a cell inside a hypotonic environment, it means that the environment has lower salt content now, and then your cell now has higher salt concentration. And what is osmosis again? Movement of water from a region of what low concentration to a region of what high concentration. And when this happens, the cell will now what start swelling up because water is moving in. Water is moving in. The cell will now what start swelling up and will eventually what rupture. Is it clear? Shall I explain further?
The point now. You said isotonic, hypertonic, and what? Hypotonic.
Yes, exactly.
Hypotonic, low concentration of salt.
Beautiful. Yes, sir. Yes, ma'am.
All right.
Any other question?
Now, please don't be offended. Can you explain that isotonic? Let me get it clear.
All right. So
Like,
isotonic, iso. Now, the word iso means same. Hmm? The word iso means same. So isotonic concentration means same salt concentration. When okay, let me define diffusion for you now. Diffusion means movement of molecules, solid molecules from a region of higher concentration to that of lower concentration until equilibrium is achieved. That means when equilibrium is achieved, there is no movement again. And people they always told us in secondary school that osmosis is the opposite of diffusion, right? So in osmosis, water moves from what region of higher concentration. Sorry, water moves from region of lower concentration to a region of what higher concentration, still until equilibrium is what achieved. Are you guys with me? So when equilibrium is achieved, it means that the concentration of both sides are now the same. That is isotonic. So when there is what an isotonic environment, there is what no net movement. And when there is no net movement, what happens? The cell is balanced. The cell will survive. And I use red blood cell as an example because that's a very popular exam question. They will tell you red blood cells, when placed in a hypotonic solution, what will happen to it? Can someone answer?
Yes. When it's placed in hypertonic environment, eh?
Yes. Yes.
There'll be movement of water from the cell to the region of a higher concentration of salt. Then the red cells will shrink, a
Beautiful.
condition called plasmolysis.
Exactly. Very correct. Very nice. Wonderful. So can someone else attend what happens when the cell is now placed in a hypotonic environment?
Water content moves into the cell.
You know it's not that. So it should be plain. You are saying, Joshua, you are saying something.
I thought someone was answering the question. So, when a cell is placed into a hypotonic solution, water contents from the surroundings move into the cell, and then the cell becomes turgid and immobilized.
Beautiful. That, so when there is what influx of water into the cell what swells up, becomes turgid, and eventually ruptures. Now, for blood cells, hemolysis is called hemolysis in red blood cells. Hem, Heme, right? Hem there means what blood. Okay, no, not that. So it's called what hemolysis. This lysis there means ruptured, breaking. All right. Okay. Now, what will happen if a cell is placed in an isotonic environment?
So, when a cell is basically placed in an isotonic solution, since the concentration of the cell is the same as the concentration of the surroundings, not the system will be at equilibrium, nothing will change.
Thank you guys. Any other question? Any question?
This one he
All right.
asked, say whether
The
he
one.
got it right now.
Okay, yes, very correct. Okay, when you place a cell in an isotonic environment, there is no net movement of water. Why? Because the concentration inside is the same as concentration outside. That is, equilibrium has been what achieved. So there is no what net movement. All right. Now, equilibrium, for water to move in or out, there need to be what a concentration grade. No, let me not introduce a new term, so we'll not get confused now.
Who told you that
So
we don't? Write
okay,
down what you are saying.
okay, all right. Osmosis, diffusion, any type of passive transportation can only take place when there is a concentration gradient. That is, gradient means difference, right? When there is what a concentration difference. Imagine inside we now have 3 concentration, outside we have 2. When you do 3 minus 2, there is what 1 deficit. Water will move from inside to outside. So when there is what 5 outside, there is 2 inside. When you do 5 minus 2, it is 3. Water will move what from inside to outside, right? So all these passive transports take place when there is what a concentration gradient, all right? Now, if there is no concentration gradient, that means the solution are what equal. We have 2, let's say 2 units of sodium chloride outside, sorry, sodium ion outside and 2 sodium ion inside. There is what, let's just use that as our source concentration now. There is what equal balance. There is a balance there. Sorry, there is a balance there, isotonic. There will be what no gradient. 2 minus 2 is 0. Are you with me? So since there is no concentration gradient, there will be no what net movement. All of us are clear. Any other question? I'm so sorry for overshooting our time this night, and I hope we actually learned a lot. We have actually started biology, and to the
A
glory
lot.
of God, biology is very bulky. I'm hearing you. Do you have a question?
This last one you explained, did you say that there is no concentration gradient?
Yes, exactly. In isotonic solution, there is
Yes.
no concentration gradient. Yes.
Okay. Thank you.
Okay, so I think this will be the end of the class for today. Please try as much as possible to invite your friends, to invite anyone you know that is missing out a lot. And you yourselves, please don't miss any class because we'll be following this closely so that all of us will go along. All right. So good night, everyone.